Hi everyone, my name is Lindsay Schwartzkopf with Beckman Coulter and I will be your facilitator for today's webinar. I would like to introduce our speaker for today's webinar, John Snyder. John is a staff systems engineer with our integrated solutions team here at Beckman Coulter. John has been with Beckman Coulter for 15 years, starting out as a software developer for 6 years and working in our integrated solutions group for the last 9 years. John has a strong background in software development, which gives him a unique perspective on our robotic systems engineering. In his role as a systems engineer, John is responsible for specifications, development, documentation, and testing of custom laboratory automation solutions. In his role, he acts as the internal voice of the customer and the technical lead of multidisciplinary teams, coordinating mechanical, electrical, and software development based on the customer's needs. Today, John will be speaking on our automated assay optimization, or AAO. His presentation will cover what we mean by the term AAO, a peek at how the mathematics works behind the scenes, Beckman Coulter's approach to AAO, and several practical examples with results illustrating the benefits of AAO and design of experiment. A short question and answer time will follow, though questions are welcome at any time. So with that, I will turn it over to John.
Thank you for the introduction, Lindsay, and we'll get started. So in our case, when we talk about automated assay optimization, or AAO, we talk about a unity of 3 different disciplines: biochemistry, statistics, and robotics. We need the scientific domain to give us some ideas of what kind of targets are we looking for, how do we measure these things, and what are optimal conditions supposed to look like. The statistics realm helps us to perform the design of experiment and do any analysis and modeling that we need to do afterward. The robotics helps us out with the transfer of liquid and does that in a random pattern so that we avoid any biases. So why would we want to optimize? It may be that you're developing a new process. It may be that you already have an assay in place, but you need to make it more robust. You might be porting an assay from a lab bench to automation, or perhaps you just want to improve your signal-to-noise ratio to make it a little bit easier to detect what's going on. There's really no end to what types of things you could optimize. Some examples might include cell media, assay miniaturization, new assay development, things like that. One of the methodologies, and the one we're going to talk about today to do optimization, is called design of experiments. Traditionally, there's no S on the end, but it's easy to throw that in there. It's also known as DOE, or statistical design, or statistical experimental design, lots of words. But basically, this is a tool that's been used quite a bit, very prevalently in the automotive industry, to try to test several variables at the same time and kind of see how those work and see the effects. So doing this type of thing, you tend to have a software package that helps you do the design and helps you analyze the results. We'll get into a little more details on that later. But the really important piece of DOE is that we can measure the effect of not only individual factors, but also detect any interactions between them. So that if it's a combination of things that are affecting your output or your result, then we can find those and optimize them. Typically, you're looking for what critical components there are in a system, but sometimes it's just as important to find out what's not critical. If something has no effect, you shouldn't use it and waste time and money on that. So starting out, we're going to have a little bit of terminology just to make sure we're all on the same page here. So we have experiments, which are controlled activities that result in a quantitative measurement. So for example, maybe we're popping popcorn and we want to see how many kernels did not pop. We have things called factors, which are things that are controlled in an experiment, and we're studying them for effect. So an example might be how long to heat the popcorn. A setting is a state that a factor can be set to. So if we're looking at the time of heating, it might be something like 3 minutes. We sometimes call these levels as well, so that's the number of settings for a factor. Maybe if we look at 2 levels for time, it would be 3 and 5 minutes. Then anything that we can't control for whatever reason, or we choose not to control, we're going to consider noise. So an example of this in our popcorn example might be uneven variation in heating from a microwave oven. So our goal in our example is to figure out what affects the number of kernels that don't pop. We're going to study 3 factors: the time it takes to heat, and we'll do that at 2 settings of 3 or 5 minutes, the temperature of high or medium, and brand X and brand Y of the popcorn. Then if we do all of those different combinations, we have something called a 3-factor, 2-level, full factorial design. So that's a mouthful, and basically it's just a full experiment. So by doing all 8 of these, we can see what has the biggest effect. And as I mentioned, sometimes it's also important to know what doesn't have an effect. If brand doesn't matter, we'll buy the cheaper one. Then we can also look at interactions. So an example of an interaction might be that cooking longer helps, but it only helps if we have the heating set to high, whereas we can cook long time on medium, but it doesn't cause any more kernels to pop. So looking at all 3 factors at 2 levels, we have a 2 to the 3rd, or 8 different combinations of settings. And we visualize this with a cube, where the vertices of the cube represent experiments. So the vertex labeled A, for instance, is where we've left everything at the low setting except for factor A, and we've set it to high. Likewise for B and for C. Then the combinations are multiple letters. So the far vertex, A, B, C, is where we have set all 3 factors to the highest setting. The experiment that is labeled with I is where everything is set to low. So that would be our kind of minimal variation. So if we want to measure the effect of a given factor, we can run all 8 of those experiments and estimate the effect of that factor as follows. So let's just look at C as an example. So we're going to measure the effect of running experiments where C is set to high versus those where C is set to low. So it's kind of looking at these 2 planes on the sphere, or on the cube, top and bottom. So the average would be something like the C minus I plus the AC minus A, etc. We go through all of those combinations, and then we average those effects together, and that's kind of our average effect that we would attribute to factor C. So we reduce that to a nice little formula, and all the other effects can be found in a similar way, and you can do the math to find out how to do interactions as well. Now, doing all of the combinations in that 3-factor, 2-level experiment isn't such a big deal. There's just 8 of them. But if we want to look at 15 different factors, which is not that unreasonable for some chemistry, that turns into a lot of experiments, even at just 2 levels each. Likewise, sometimes we would want to study more than 2 levels. So you can imagine having a 3 to the 15th, etc. So what happens if we only do a fraction of those? Well, that's what the field of fractional factorial designs is all about. And there are other types of designs as well, but this is the one we're going to look at for an example. So by picking the quote-unquote correct half of the experiments to perform, we get a nice design. So looking at our C effect here one more time, just looking down the column, you would see the minus sign for everything that we had subtracted earlier and plus sign for everything we added. But now we're only doing half of the experiments. Those are represented by the diamonds. So we only get to look at half of the rows because those are the only results we will get. When we add those together, we get a slightly different formula. Notice that the column right next to that, the AB column, looks about the same. So those 2 things are said to be aliased, and that's just the price you pay for not doing all the experiments. Also part of DOE is the ability to make a prediction model. Now this is a mathematical model. It's not necessarily related directly to the science involved, so it's not biology or chemistry. But it gives you an idea of what the math is telling you. And for a localized area, it can give you a decent predictor of what's going on in your system. The model depends on the experimental results, and you're going to test the null hypothesis of a factor not having any effect by plotting the effects versus any expected noise. Things that look like noise would fall on the line of a half-normal plot versus those that are a little bit off the line or a lot off the line are those that are not similar to noise, so those might be significant. So after we pick those things that look like they might be significant, we let the stats package do all the hard work, and it builds a model for us. So for our example popcorn experiment, we might have something like this. Y, which is the kernels that are unpopped, would be an addition of all these different things, where the negative 6.5 is the estimated effect of A, the minus 20.1 is the estimated effect of B, etc. And it's just a linear model. So this shows, for instance, that the time of heating and the temperature are both important factors for our popcorn, but also the time and temperature have an interaction effect, where having them both set to high reduces the number of kernels that are unpopped, which in our case is a good thing. So there's an entire field of mathematics surrounding this called statistical experimental design, and it extends these ideas to many factors, many levels, many different kinds of designs. So you can look at it at a far more complex thing than what we just did, but I wanted to give kind of a quick intro there. Here's some additional types of designs where you might pick some extra points that are outside your cube, plus one in the middle, or you might pick some that are on the face. That's called a face center design. You might pick some that are on the edges of your cube, or you might even get something kind of esoteric. I never would have thought of this myself, but there's the type of design that you could do. I think it's readily apparent that you want some software to help with this. And there's really kind of two pieces of software that are involved. One is helping you with your statistics design and analysis, and one that will run the robotic pipetter to control it for all your liquid transfers, speed that up, and make it less error prone. So this is probably a good time to take a break and see if everybody's following okay, if I've gone too fast or too slow. And I don't know everyone's background, so I didn't want to leave anyone behind. If this is repeat, I apologize.
So if anyone has any questions, if you wouldn't mind just typing those into the chat at this point. Or if there's no questions, if you just want to let me know and make sure that everything's still going along okay, that you're okay to continue, then we can just move along.
So we're not seeing any questions pop up. I think I'll continue a little bit.
Okay. Thanks, Christopher.
That was just a quick introduction to what experimental design is about and kind of a little bit about how it works. It's not really necessary to understand that in order to use this, especially if you have some statisticians helping you or if you already know a decent amount about DOE. So I wanted to talk about the software that we have. We ingeniously named it AAO after the Automated Assay Optimization. This is kind of the opening screen. Basically, this software is a bridge between the mathematics and the robotics. You use it to import your experimental design from your favorite statistics package, and then it will create a randomized plate map, automatically create any liquid handling protocol that you need, and then you collect the data any which way that you need to for your assay, say with a plate reader, and then this can read that in and deconvolute it, by which we mean it undoes the randomization of the plate map, so that things are now in a nice order for your statistics package to do analysis. So here's kind of a graphical overview of that process. You start in your favorite statistics package, create the design that you need, import that into our software, which then makes the method or dispense table that you need. So in the case of the BioRapTR, it's going to be an Excel file in a proper format to be run, and it will either use 4 or 8 bottles depending on your configuration of the device. Or if it's a Biomech FX unit, it will control the pods on there via a regular Biomech method. We have a little bit more control there because we're able to do a wider variety of transfers than we can with the dispenser. But it's your choice as to which instrument matches your need. Once you're done with the assay, you'll spit out data. I just have a spreadsheet representation there. That data is then imported back into our software just briefly so that we can undo the randomization. And again, that's to help avoid any unintended effects, such as side effects of plates or systemic errors, things like that. Once we have things unrandomized, you put it back in the stats package and it does the analysis. So just as a quick introduction for those who may not have seen the Biomech FX, it's a flexible liquid handler, has a very large capacity deck, and has 2 different pipetting pods, a multi-channel and a span 8. The span 8 is capable of addressing a single well all the way up to 8 wells simultaneously, and the multi-channel does either 96 or 384 wells all at once. So the multi-channel in this case would be used for adding bulk reagents, not typically for factors that you're studying. That's what the span 8 would be used for. The AAO software for BioMech-FX creates and validates the BioMech methods, and then utilizes the span 8 for the randomized liquid handling. It can also do scheduling of per well incubations. So if you want to study time as a factor, say for a stability study or something like that, this makes it fairly easy to do. And the BioMech-FX can address either 96 well or 384 well plates for this solution. Moving on to the BioRapTR, it is a rapid non-contact multi-reagent dispenser where each channel corresponds to a liquid bottle and has its own independent fluidic pathway. And it can do from 100 nanoliters to 60 microliters per dispense. So this is a really nice unit if you're studying miniaturization of an assay, or if you just know that you want to move that direction and save some cost on reagents. Also, it is very fast. It can do an entire 1536 well plate with 8 reagents in less than 5 minutes. So this really makes DOE a very rapid thing to look at. The AAO software that runs the BioRapTR generates the dispense tables, which is an Excel file, and can address either 384 well or 1,536 well plates. So I'm going to switch to showing the software just briefly. There's kind of 10 steps that you go through when using the software. One is to import your statistical design, whether you use Design Expert, Jump, Minitab, Statistica, whatever package you use. Or you can create your own in Excel, save it as a CSV file, and we can import that. Once we import the design into the software, it can be used to do design parameterization. So this is where we will pick which things in your import file correspond to which factors that you're studying. Or if you have a blocking factor, you can do blocking across plates or parts of plates and things like that. The next step is then factor type specification, which is either a set of 2 things or a set of 4 things, depending on the unit you're using. The screen capture here shows just 2 factor types, and that's for the BioRapTR. So either things are reagents that you're dispensing, or you're studying something that varies from one plate to the next, and we call that an interplate factor. On the BioMech-FX, we could also study incubations, or we can create what we call buffers, which is kind of a misnomer, but basically is a mixture of any reagents that are stable enough to be mixed together ahead of time and then dispensed later. The middle step here is to select a plate type, which is basically just what size, and do you want to restrict any well that could be used. So for instance, you may want to avoid the edge wells because you know that those have an effect, or perhaps you've reserved a column or a row for quality control and you know you'll use that manually. You want to avoid using it with automation. Step 6 is how to do randomization. There are 3 different possibilities, and basically this is responding to customer requests. So we have the ability to do any sort of randomization you may desire. The default is probably the best from a mathematical point of view, but may not fit your processes exactly. If you don't want to, you don't have to use any of these randomization options, and you can instead specify where all of your experiments are going to be in your import file. So we allow you to specify well locations for each and every experiment if that's what you want to do. Most people let the software do this for you. Step 7 is rather automated and just generates a randomized plate map. You can see one of the pretty pictures from a 384 well plate there. Really, this is just to give you a visualization of how the randomization is done and to kind of confirm as the last step before making methods that you have all the factors set the way you want with the different levels that you want. Step 8 is to generate your liquid handling protocol, which will either be a BioMech method if you're going onto the BioMech-FX, or it will be an Excel file for a BioRapTR dispense table. Step 9 is to run your protocol, which is done the same way you would with any assay. But I wanted to interject a note that these are both fully editable. So if you decide when you've generated your biomech method or your dispense table that you want to make adjustments, you can do that right then and there. You don't have to use our software to do that. So you can add an additional reagent, reorder things, add additional actions for the robot, whatever you need to do. After running, you collect the data and then import it back into the software. This is where the software will do deconvolution, and you can then copy-paste or move that data into your stats package, however it works, for analysis. Normally the easiest way is to copy-paste from Excel. So that's really all there is to using the software. There are a few additional configuration options. I mentioned that you may have non-experimental reagents. So if your assay always needs a buffer or a diluent or something like that, you're not studying that thing, but it has to be there, you can add it. You can also create and assign randomized control wells, and we can let those be either a total well that's doing essentially a full quality control, or you can do positive or negative controls depending on what it is you're doing. Or you can actually mix all three of them together, and those will be randomized across the plate just like a regular experiment would be. So that's kind of nice for making sure that your assay is working properly. Additionally, on the FX version, you can specify per plate or per well incubations, and you can specify the order of multiple assay steps. And these can be within a single method, or you can generate multiple methods. There's no reason to restrict yourself. You can do it however it makes the most sense for your assay. So this might be a good time for another break to make sure everybody's keeping up okay and see if we have any questions. Or if you have comments about the pace, if I'm going too fast or too slow, that's fine too.
Okay.
Not seeing anything, so we'll move on. So some of the examples that we have are collected from various places. One of the examples is from a high-throughput screening assay optimization that GSK did some time ago. They were monitoring the results of applying AAO, not just applying it, because they were an early adopter of this technique. So they knew that it would work, but they kept applying it to different assays. And as time progressed, they kind of collected some additional information. So among their development goals are to improve their signal-to-noise ratio, improve their robustness and automation, and reduce their assay volume and cost. Some of the results were rather astounding. The goals were met across the board. And in particular, the signal-to-noise ratios were very, very impressive. They had one radioligand binding assay that had as bad as a 2-to-1 signal-to-noise ratio, but it was highly variable, so you couldn't rely on your results from one step to the next. After optimization, it was stable, and they got a 20-to-1 signal-to-noise ratio. You can read through the table there to see some of the other improvements. They also had an enzymatic assay that was catalytically bound, and they had trouble getting it to run in anything less than about 16 hours. And they were able to improve that by 4-fold. And so they could run multiple assays per day instead of a single one overnight. Another benefit, and they got this by tracking several applications of AAO, is that they saw a cost reduction via the speed of their optimization. The timeline for doing an assay optimization using their traditional methods were generally in the 4 to 12 month range. They could do between 1 and 3 iterations of assay optimization using AAO in 4 to 6 weeks. They estimated that the savings on time and reagent at between $10,000 and $40,000 per target. And I wanted to note here also that most of the time the iterations were set to 2. Sometimes a single round of optimization was enough, but frequently they would do another round just to get things really tweaked in. And it was very rare that they had to do this 3 times. Another example comes from multiplex PCR optimization. This is where our scientists were studying primers from a Weber panel for genotyping human DNA, and the regions of the DNA are amplified, by these primers, and they're looking for, or sorry, they are short tandem repeat regions, or STRs. So these are what I guess the scientists would call an STR primer, and they're trying to figure out how to multiplex these, but combining multiple primers in a single PCR is kind of tricky. There are a lot of things that can have interactions and influence your results. So without a good design behind what your experiments are, you can get lost in analysis pretty easily. So some of the goals were to optimize the concentrations of 3 pairs of STR primers, and then also magnesium chloride on a CEQ genetic analysis system. When doing this, they should get 3 distinctly sized markers that represent an individual specific genotype. And then they also wanted to look at the effect of small changes in annealing temperature. Basically, how well do you need to control your annealing temperature and still get good results? So here's a before picture that shows the results prior to optimization. The signal that is seen below about 135 is noise, so ignore the 131 peak. The peaks they were looking for are in the 139 to 159 region, the 189 to 193 region, and then again at the 210 area. Notice that that's a very, very small peak. The signal's very weak, but the software did call that out correctly. So it's good that it found it, but they wanted things to be much more robust. After optimization, we get a much better signal. So we still have the noise down around the 130 area. The black and green areas there in the 140, 160, 190 areas are still very good, but we were able to bring up the signal in that 210 area to be quite robust. So that was a pretty significant result. In the end, our scientist was quoted as saying that their assay quality was significantly increased without sacrificing resources and precious samples. As a side effect, they also determined that even a 10-degree change in their annealing temperature had little effect, which is a rather sizable change and not expected. But it's a good thing for them because they don't have to watch that quite as closely and don't have to control it as well. So
sure. And so everyone's also unmuted on your phone, so if you have any questions, please feel free to jump in at any time.
All right, so another experiment that was recently published was an optimization of embryonic stem cell differentiation into cardiomyocytes. So we're studying combinations of what our scientists coined pro-cardiomyocytic compounds. Basically the things that would help create that differentiation. So he did a literature review to try to learn of all the different possible compounds that people are trying. And then he wanted to look at concentrations of those compounds, the time of addition in the process, and the order of addition to look for optimal conditions for creating cardiomyocytes. An interesting thing about this experiment is that he had no prior knowledge of what the effects of these combinations might be. He didn't find in the literature where people were doing combinations of most of these compounds. So a differentiated cardiomyocyte cell will frequently start spontaneously beating. I don't have a video with me, but I have a region of interest outline of what is seen as a beating muscle area as seen under a bright field microscope. So you can kind of get an idea of how much of the area of these cells is actually spontaneously beating. For measurement, though, we wanted to stain things and be able to run them in a way that we could count. So just looking at it visually wasn't a quantitative measure. So some staining was done with the Alexa 488 conjugated antimycin heavy chain antibody. And when you look through a fluorescent microscope and take a picture, you can see that very similar regions to those detected as beating visually are also where the fluorescent tag attached itself. And we're able to see how that correlates. So this is something we can measure. So when we run those stained cells through the FC500 flow cytometer and then analyze them with our Kaluza software, we can see that the undifferentiated cells are all below the gate. And on the other side, you can see that only a few, around 6% of the cells, are showing above the gate. And that's okay. That's not a bad result, but it's not what we were looking for. So our scientist set that percentage of positive cells as his measurement for this optimization. So again, kind of zoom in on the control set. And I'm sorry that some of those red dots aren't showing up for some reason. Hopefully they show up on your end. But basically, we're going from about 5.9%. Through the application of 2 rounds of AAO, we're able to get to 43%. So that's a pretty significant change, about a 7-fold increase in the number of cardiomyocytes that we got from our stem cells. If you're a visual person like me, you also want to see what does it look like under a microscope. So here's the region of interest for the control when we differentiated the cells, kind of using the quote-unquote standard recipe. With the optimized recipe, we get much larger regions of beating cells. And since I'm not a scientist, rather a computer scientist, I'm more gee-whizzed by this than many, but I thought this was a very neat result. So I also have a few examples of how experimental design, or DOE, can be used, not necessarily in conjunction with the AAO product. So this is just trying to show some of the other areas where we've used this. One is with our high-density replication tool, where we're doing liquid transfers by dipping pins into a source and then the destination. And then we have to wash those pins between transfers to avoid cross-contamination and to produce very precise transfers. These are on the order of 50 to 100 nanoliters. Sometimes you can go even lower. The wash is done by dipping the pins into multiple wash solutions and then sometimes drying them with a fan. Our goal here was to increase the precision after a wash and also minimize the amount of time spent doing the wash. By using AAO, in just 4 business days, we were able to find a good set of parameters producing acceptable results. We later found out that an expert in the field, one of our customers, had come to very similar conclusions on washing parameters, but he did not use AAO, and it took him many months to get the same result. Another area of using DOE was pipetting optimization. Back when we launched the Biomech FX around 1999, we were challenged to try to come up with a characterization of what this new instrument could do. We always want to do smaller volumes more reliably. And a quick analysis showed us that doing 1 factor at a time, or OFAT, would have taken way too much time and taken way too many resources through tips, reagents, etc. So we applied DOE to the problem. It was an effective way to understand what was affecting our pipetting results and to minimize our resource consumption simultaneously. Initial testing, just using the out-of-the-box pipetting technique, gave us a CV of about 3.6%. After optimization, we were able to get 1.7%, which is better performance than we'd ever seen with Biomech FX. So just another example of where DOE can be applied. In conclusion, we found that DOE is an effective tool for experimentation. There is plenty of good software out there to help facilitate its use, both with and without our liquid handlers. There are resources available to help use DOE, whether it's a statistician or a robotics company such as Beckman Coulter. And DOE is really helping to save resources and provide better solutions in a shorter amount of time. So our coin phrase is to do experimentation faster, better, and cheaper. I have some references available for anyone who's interested in some of the journal articles that we've seen over the years. And with that, we're ready for questions. And thank you very much for your time.
We did have one question come in. Fred was asking, were the stem cells added using the BioRapTR?
I believe so, yes.
Okay.
Yeah, the BioRapTR actually is a very good dispenser for cells. I don't have the data in front of me, but apparently it's better than a lot of other techniques.
We'll give everyone a few minutes to see if you have any questions. You can type them in the chat, or we should be able to hear you if you would like to speak your question as well. And and Fred was just asking, can we go ahead and remind what is required to use AAO?
Okay, so for the AAO software, you'll need Biomech software, even if you're using a BioRapTR, because we use its library of labware definitions to kind of keep track of volume tracking and things like that, and some of the other internal tools. So Biomech software is required for either case. In the Biomech realm, we would need a hybrid unit that has a multi-channel pod and a span 8 pod. That has some side effects of needing a tip loader, a tip wash station for the span 8, and various deck positions just to put all of your reagents and tips on. If you're using fixed tips, you use less space on your deck span 8. However, if you use fixed tips, you're spending time washing. So you need to make a choice there. On the BioRapTR, you can use either the 4-bottle or the 8-bottle version. They both work interchangeably. You can do experiments with more than 8 bottles. Even on the 4-bottle version, there's no restriction. We give you the option as a user of the software to pick how you want to group your experiments. So you can do either 4 or 8 combinations at a time in batches, and then replace your bottles, clean the lines, and do another batch. And then, of course, you'll need the AAO software. You'll need some sort of statistics software, or if you're comfortable creating them yourself in Excel, you can do that. I recommend having statistics software just because it helps direct your experimentation and keeps you on track. I think that's about it. We do use Excel pretty extensively as a means of data collection and kind of a quick way to turn files around.
What is the maximum number of variables that can be evaluated in one experiment?
Maximum number of variables. There are practical limitations that will get in the way before the software will. The software will let you use any number of experiments, any number of factors, any number of settings. So you can go up to really crazy things like doing 30 different factors all at the same time in 3 different levels each. And we have customers that are doing that. That becomes kind of tricky on the analysis side, at least partially because there are a lot of interactions, and you want to be able to kind of tease those apart as you do your analysis. Most analysis software can handle up to about 30. There are some packages that are limited to 15 or so. So it kind of depends on your statisticians and which statistics package you're using. I know that Design Expert will let you go to 30 variables. We typically don't recommend going over about 15 because it gets to be just too much to keep track of. But if you're comfortable doing that and have good support, go for it.
Thanks, Sean. We'll wait another couple of minutes and see if we have any other questions that come through. Okay. No additional questions at this time, so thank you for presenting, and thank you everyone for attending today.
And if anyone has follow-up questions, feel free to send an email maybe to Lindsay as kind of a clearinghouse, and she'll get those to either myself or whoever at Beckman can answer them best.
Or to Fred as well.
Right.
Okay. Thank you, everyone.